All right, so like many people here, I'm really interested in sort of in cognition and in particular decision making. And my the way I got interested in this problem originally is that I sort of want to understand how groups make decisions to work together. And the way I started thinking about this at the beginning was that I thought about how do people make decisions, right? And I sort of thought loosely there were a few different paradigms of how people make decisions together. We sort of have a follow the leader paradigm where like a king or maybe a department chair or somebody tells you you need to do X and you do X sometimes to varying degrees of efficacy. We have maybe a group discussion or consensus building paradigm. Maybe you have a committee or Congress is trying to decide what to do, and that also sometimes works well and sometimes does not. We also engage in sort of behaviors where it's sort of hard to say we have a conscious decision, but these sort of collective behaviors like maybe being part of a stadium wave or a mosh pit where we all sort of know what to do. There's almost a program, we see it, we participate. Right, those are sort of the three general ways I see us as deciding let's do something together. And originally I thought maybe cells have a similar way of thinking about things, right? In your heart you have cells, pacemaker cells, that tell the other cells in your heart to beat, right? You have 1 in 100 of them, and if they're not telling everyone else to beat along, you have a problem. There's also lots of beautiful work on, say, epithelial cell sheets or other cellular systems where it's very much like a collective behavior kind of paradigm. There's some sort of physical movement, the other cells feel it, they move in concert. You can sort of think of it very much like a very basic physical system, just like our stadium pits and mosh pits. But I want to understand, do cells have a sort of group discussion paradigm to execute more complex decisions? And as I sort of tried to break this down a little bit more deeply, I came to the conclusion that basically I can sort of characterize each of these bins and tell you that cells do some of these things and not other things. But something really jumped out at me, and that's that all decision making, actually with us, even or with cells, requires individuals respond to local information. That's a really key unifying feature of having to make a decision. All right, I'm making a statement here that we don't have something like telepathy, right? It's not enough to think on the other side of the planet that you'd like me to make a decision with you. You have to communicate that information to me somehow. Right, that might be through an email, there might be some sort of long-range communication that I receive, but I need to receive it at myself. All right, and cells have a similar kind of need in order to work together and make decisions. All right, so local information to cells can look like things like mechanical forces, maybe biochemical signals or pre-established gradients, electrical signals and gradients, all sorts of things, right? But they have to be able to receive it at them to do something with that, right? And they can have all sorts of mechanisms then for inside themselves interpreting that information and making some sort of decision about how to work together. Right? So this could include things like maybe you have a stochastic switch. I see something, I can do A or B. Maybe I have a threshold detector. I see a signal is on. I will execute a program. Maybe I'm integrating a signal over time. Right? But there's sort of a bunch of different ways I can do this. Right? And it's these individual cellular units that are interpreting information from their environment that build up more complex multicellular behaviors, right? So cells take this environmental information, they encode it, and then they do things with that information that are the building blocks for larger scale behaviors that, say, a group of cells or even a whole organism might do. So a single cell might do something like secrete a signal, it might move, maybe it'll turn on a gene expression program, right? And multicellular behaviors are basically built up of these single cell decisions and behaviors, right? They may relay a signal across a whole population to synchronize something. Maybe they'll migrate as a whole group, right? And it's these multicellular behaviors that basically the building blocks of emergent behaviors like development or tissue repair, right? That are really key when a group of cells as part of an organism needs to decide to respond to something like stress in nature. So that was thing number one I thought was really important for how cells behave. The other thing I thought about how cells behave is it's not just important to understand the local environment, but if each of these cells has different ways inputs it can receive, and a particular program that's tuned to those inputs, we might really care about the kinds of cells in the computers we're looking at. Right, so we often look at cells in the laboratory in a dish, in a laboratory kind of environment, and think how do we sort of normalize everything and make it all the same? How can I easily propagate this organism, make it uniform? But native cells have existed in a totally different universe where they're used to receiving somewhat different inputs from a very different environment. Or the environment, in fact, looks very different. It may not be sufficient to take those cells and put them on the laboratory kind of environment. And it may not be sufficient to take lab cells and put them in a naturalistic environment. That the real goal is to ask, how do cells compute in their natural environments? Right, so this is something maybe in vivo researchers will think, oh yeah, of course, I do, say wound healing in an animal, that's much more relevant than wound healing in a dish. That may in fact be the case. All right, but it's something we struggle with, maybe even animal decision making. What's the equivalent of understanding how rats behave in their native environment? Are we really recreating that in the lab for decision making? So, but the question I was asking is sort of, how does this work for cells? And the model system that I think about is the cellular slime mold. So just to differentiate it from the system you were introduced to this morning, this is a group of many different cells, each with their own nuclei and cell walls. And when they run out of food, they make this beautiful structure here. All right, so you might imagine this out in nature. You're going on a hike, you look down, and you see on a piece of mulch these little structures, a few millimeters in size. These are cells that have run out of food into the soil, starved, started telling each other, hey, we got to get out of here, we got to go somewhere else, crawled out of the soil, and gotten up on this piece of mulch to hang out somewhere where they can wait for more food for a while. And this is an incredibly dynamic process. So I'm going to show you a video of what it looks like. And so what you're seeing here on one side of the screen is that over here, this is a lawn of bacteria. So you can use all sorts of stuff. They are pre-promiscuous, Klebsiella, E. coli, whatever you want. This over here is cellular slime mold, and this is a feeding front. This is where they're sort of eating the bacteria and gorging themselves. And as they gorge themselves and run out of bacteria, they start to turn on a program, and they're communicating with one another, and that program makes them round up. And that's why you're seeing those waves. Right, and those waves are helping them find one another and synchronize their gene expression. And they're going to end up in these groups where they differentiate roughly into one of two cell types, generally speaking. About 20% of the cells become these stalk cells, or that you saw holding up that structure, the spore structure. Right, that spore structure contains about 80% of the cells, and it can hold out for a month or so in the wild until an animal maybe bumps onto it and can carry them somewhere with more food. All right, so this is an incredibly dynamic process. It happens relatively quickly in terms of development. We're now about 21 hours in, and so these cells here that were feeding previously have now starved and built a structure. This is an incredibly dynamic, required process, right? If they do not do this, if they cannot manage to come together into this structure, they will die. It's life or death. So I want to go ahead and zoom in on what the cells are thinking about their environment when this is happening. So what I'm going to show you here is we're going to look at an intracellular signal called cyclic AMP. If I can have you remember one molecule today, I have to admit I'm not a biologist by training, so I get really bad at those talks that are like, you need to remember this whole signaling network. Remember one molecule. All right, they're using this molecule inside themselves to synchronize their gene expression and also to send a signal to other cells. So they'll send out the cyclic AMP outside of them and pass it along from cell to cell. And that process looks relatively similar to what you were seeing with those sort of rounding up waves before. All right, but let's go ahead. Nope. Come on, you can do this for me. The joys of always transferring your talks to someone else's computer, right?
You can use the computer keyboard.
All right.
Yeah.
Come on. Oh, no, it cannot play media. You can't do this to me. This is the worst. All right. Let's give it one more go. All right, tragic. This is my favorite video. So you have to imagine this for me. All right, that these beautiful spiral waves, this is crushing, it worked a few seconds ago, are emanating out from different centers. So you can see here, there's a little red dot here and a red dot here. All right, there will be beautiful spirals, and I checked a bunch of the other videos, and they hopefully will still be working. It didn't stop in the past 10 minutes. And they're driving all of the cells to come into these mounds where it continues to spiral around. All right, and so they're using this for a few different things, like I said. The first one is to coordinate gene expression. Right, they're going to do the initial stages of becoming either a spore cell or a stalk cell. And we think we understand roughly how this whole thing is driven. Right, so people have been thinking about this signaling network that drives these spirals for decades and built models of how cellular signaling networks must be constructed in order to make these patterns happen in 2D to drive the initiation of fruiting bodies. And I thought what to do today, and I thought, well, I could dive in on the math and give a talk similar to our earliest talks about, what are the sort of requirements for cells to process information. And I thought, this is a conference, and the fun thing is talking about data. I can't just point you to a paper. So to summarize, both some work from my lab and some other labs of the past about decade, people think they really understand now how cells that operate in 2D have to process information to drive this process. So we think Each single cell itself is excitable, and they almost perfectly adapt to changes in the environment. They exhibit fold change detection, by which I mean they're really good at being detecting order of magnitude changes, and basically the relative difference in the environment is really important. Sometimes there's a sort of ongoing debate. We think that the sort of stochasticity is really important in their ability to process information as a group, and that over time that they get more and more excitable, and that's really important for them to find each other and drive this process. But something I really wanted to understand is that, again, this is a sort of 2D laboratory environment, but in nature, this whole process has happened. This whole process has evolved. These waves, this whole network, evolved in some 3D heterogeneous environment. So what part of our understanding actually applies to this process when it's happening in the soil? All right, how do you look in the soil? Like, I can't just stick a microscope down in there, right? I can do endpoint studies. I can see that cells come out, right? It seems to work relatively well, but what's going on in there? So this is really a challenge. My lab does both theory and experiment, and my joke to you, seeing is believing. You shouldn't believe me that those 2D waves even happen. You should come find me later and I'll play you the video. But I really did want to understand what are key features for naturalistic soil that I can actually look in to see what's going in there to understand how environments shape behavior. Right? I need something that is both compatible with my ability to look in there and quantify behaviors, but also, I can still say this actually mimics an environment and allows me to quantitatively probe how cellular decision-making works. All right, so when I think about soil, fundamentally I think about it as particles of different sizes. All right, and if you think about the world from the point of view of a cell, and if local interactions in your local environment really matters, there's sort of 3 regimes. All right, the first regime is that you have cells, and the particles are similar to the cellular size. Think of this as like silt. All right, you can model soil as sort of a defect in their network. And as the particles of soil get bigger, it sort of creates channels and irregular regions cells can move and signal through. And as they get even bigger, the soil creates large irregular regions like rooms that have really interesting properties that are different than you would get in a 2D system, because you have surfaces that cells can be on. These cells don't swim, so they can be on the rocks or on the bottom, but they can't jump from rock to rock. That said, they can signal and send signals if there's liquid media between them. All right, and so you have these very different kinds of spaces now with very different properties that are going to affect your ability to transmit information, as well as your ability to interact with your neighbors and make decisions about how do I get out of here. All right, so to visualize decisions for information processing, what else does synthetic soil need in the lab? So it needs to be transparent. I want light to be able to go through it, but I also don't want light to hit every interface and bounce off. And so we need something that's refractive index matched, so I minimize scattering in my sample. I want to be able to visualize where the particles are. I want to visualize the cells. Maybe I want to visualize their signaling behaviors. I want something that's survivable, so this is something with a good pH, it's aqueous, has oxygen penetration, good humidity. And I also want cells to be able to grow and move in these environments. So the solution to this problem turned out to be a mineral called cryolite or ice rock that people used to use way back in the 70s or so for studying marine sand and how marine organisms burrowed. And it's actually sort of a fun story. People in the lab couldn't get it anymore for a while because it's used in industrial processes. For a long time it was the source of aluminum for a lot of industrial processes. And then people found an alternative in industry, and it became available again. You can buy this on Etsy. They claim it's a crystal that has good vibes. So my joke is that I started working with this in the lab, and then I got my dream job, so that was really great. Highly recommended. That said, just before I got my dream job, I was reading this paper about how it was being used to look at bacterial and fungal growth on tree roots. And I thought, oh, well, if bacteria will grow in there, Dicty eat bacteria. I wonder if I can grow Dicty in there. That would be great. So it turns out it works. So what you're seeing here is a small little microenvironment, a few millimeters high. What's glowing here is the liquid, and then you can see each of the rocks themselves, just to get an idea of particle size. So we have really great control over the size of the particles. Basically all you do is grind up the mortar and pestle. You can use sieves to separate things out and create a custom soil mix of any kind of soil environment you'd like. You can seed cells in there. So blue is now that liquid media. The rocks are black, and the cells are yellow. We also put bacteria in there that's unlabeled. But we know it's there because if we wait over a day, the cells have proliferated. They're able to feed and grow. They eventually reach carrying capacity and start to starve. And so that's how you start to see those waves that you did not see before, but now you can get an appreciation for making these large, beautiful arcs. And those waves start to drive aggregation of the cells into smaller groups. And here's just another example where you can see with bigger particles, they'll do the same behavior, starve, and aggregate together and start to escape the soil. And just to give you an idea of, let's see, I want to sort of zoom in and show you a few different things. One, here's an example of some of the beautiful streams you would have seen in 2D that also exist here in 3D. So you can see that there's a sort of wave that propagates, there's these red different lines and ridges, and all the cells will come in and swirl and aggregate, and they seem to aggregate preferentially on these rocks. Also, just to give you an idea of what those spirals look like in 2D, you can see them here in 3D. So here's an example of a spiral and signaling the sort of channel, just showing that there's rocks all over, and yet the spiral is still coordinated in 3 dimensions across the cells. So you can also go ahead and get a sort of finer scale picture of how cells behave in this environment, and 2 different features of how they're going to work together. So here's a good example of how you can tune the density of cells and still see these kinds of behaviors where the cells are able to aggregate, sometimes more optimally than others. You'll notice at a very low density, they really struggle to get together, and I'll sort of pick on this later. But even at a high density, much higher density over here, you're going to see that we end up with a lot of different groups, and that's a thing I want to talk a little bit more about. So we can go on and go look deeper in the rocks and find out exactly how these cells are behaving at a much faster and a smaller time scale. So if we sort of zoom in, now the videos I'm going to show you are taking slices where we go up from the bottom. You can see here a good example where we've got a stream of aggregating cells, and you can see these waves propagating down the stream, driving the cells in one direction toward some sort of signaling center. And there's a real 3-dimensionality to it around this rock. So as you can see a lot of stream here, but as we go deeper, the stream is actually angled upwards. Right, it's crawling up towards something that's attractive out of our field of view. So again, just to give you an idea of what's going on there, this is the same area but at 4 different depths. And so it's also the stream is able to bifurcate a little bit around these rocks. Here's an example of a rock and sort of rejoin itself. Something I think is really cool is that the cells interact with and remodel their environment somewhat. So if the particles are a little bit smaller, such that the cellular forces are able to manipulate them easily, you see behaviors like this, where you can see the media here is moving along with the cells, so that the rocks are shaking, and that they actually are able to grab this rock and take it away with them. I don't know what's going on there. I've only caught this in the act once, but I find it really quite interesting. So just to sort of give you a close-up of what's going on there. It's not quite clear if they're getting stuck or getting wrapped around it, but they take it along for the ride. So these cells really do like to be on a surface. They're able to bridge surfaces. I'll dive a little bit into that in a moment, but they do like to be on rocks, so it makes sense if there's something that's particularly appealing for some reason, they might want it. So I made a comment earlier about how cells in the system can't swim, but you might really want but that's not true if you have a whole group. So this is a sort of side view of some of the structures they'll build in the soil. So each of the gray things is now the rocks, the yellow is the cells. And you can see here that they actually build a bridge to help them get up on top of a rock. This bridge is almost 200 microns tall, so over 10 cell heights. But I kept calling out that these cells are making small groups. And something that really struck us after we were taking a bunch of these videos is we would see some cells get out and some structures, but we were lacking sort of a whole picture, right? If you're taking a small snapshot of a much larger environment, you oftentimes miss what's going on. And so we were finally able to zoom out and get a nice set of imaging conditions fairly recently, and we watched what happened to lab strains. The following happened, and this was really upsetting for us. So you can see cells start here mainly on the bottom. They're moving around, and they're going to start to signal each other as they starve. So you start to see those red waves be transmitted across the population. And they drive the cells in, and then the cells make those little groups, and they sit there. And they'll just keep sitting there. Occasionally, a few will escape the very edges, and so those will build the fruiting bodies we were seeing at the ends of experiments. But like, this is death, right? If this is what happens in nature, you do not get out, you totally die. This is a disaster, right? It's not just enough to stick some lab cells back in nature. They totally fail. So we thought, well, what's going on? Are natural cells any better at this? Like, is something wrong with our system? First, of course, we went down the rabbit hole of clearly we're the problem. Turns out we are not the problem. If you take wild cells, so these are a little bit harder to transform, so this is only partially labeled, but you'll get a sense of sort of what the group is doing. They actually have a very different behavior. So there you can see cells on the bottom. You're not going to see signaling, you're just going to see their movement. But they really get out of there, right? They build these huge streams that are really effective of getting out of our very large field of view. All right, they know how to exit the soil. And basically, if you run this experiment over a wide range of densities of cells, wide range of particle sizes, every single lab stream we've looked at, we've looked at several labs, 2 different lab strains, as well as multiple different wild strains. Wild strains are always way better at getting out. There is something that they do that is totally different. I have no answers for you. There are many different things it could be. I am excited to chat with you about possibilities. Some of the things we've probably thought of and are on our massive to-do list. Some of the things are not. But just sort of like to wrap this up and sort of give you a paradigm. Right, decision-making mechanisms optimized for lab environments can fail in nature. Right, something about this system over time does not work in the natural world. So in 2D, we have a system where you have a bunch of cells. They signal each other. They propagate waves. They aggregate together. They'll sort out. They have a kin selection system. Everything here was clonal, but they will sort out into different groups, different cell types, and put up this fruiting body. Right? It's a little bit more complex than this. This is the sort of general paradigm that we think is the most important stuff. Over the years, people have said, well, there's some group size regulation. There's a counting quorum sensing system. People have sort of thought it wasn't really that important. We disagree. We think this might be what's driving this. There's kin discrimination, like I said, but this is not what this looks like, and we're working with clonal populations. Stickiness is really important. Once these cells find each other, they adhere. Maybe that's something that's going wrong. We're not sure. Maybe natural cells are just better at being sticky, and lab cells have lost that, right? But even if they haven't, this is already a much more complex decision-making process where there's actually many different failure points. So you have to be able to find each other in 3 dimensions. Maybe there's something about the properties of the waves that just aren't as good at building large groups, and the groups don't hit a critical size. All of these different aggregation processes need to happen where they can find each other and build a large enough group. If you can't build the right size bridge, maybe you're in trouble. You sort out. You need to be able to get out of the soil. So they have a collective motility phase that actually, if you can't move fast enough, or be able to get through this environment, you're not able to get out also, right? And then they build a fruiting body. So, like I said, we're thinking group size regulation is possibly what's going on just from the timing of when they break up. But we're also very interested in the idea that maybe this is some sort of epigenetic or other regulation of one of these systems that, we could say has a genetic fingerprint, but actually when you dig in, maybe we can select for this over multiple generations and recreate these a couple of different ways. So I'm excited to chat with you about what this could be. And with that, I want to thank everybody who did the work or helped support it, and I will take questions. If you're not here, or even if you are here and can't chat, please feel free to email me if you don't get your question in. I'd love to talk more. So thank you.
Questions, please?
So that was extremely cool. I had a question about the last part where the wild strains kind of can get out. Is that primarily geotaxis? I mean, are they basically saying, I want to go up and not down? Or is it an out, like they know that they can go to the side? I guess I'm wondering, what
are their getting mechanisms?
Yeah.
So it's been previously shown that actually they do not care about gravity. They will do this upside down. And when I do this in the lab, I actually grow my plates upside down just to prevent moisture from falling on them. They have a couple of different ways they do it. One is they're emitting ammonia, and they're sort of checking for how fast it dissipates. So there's a sort of, not quite there's also thought to be a oxygen-based taxis as well, but they're looking for, am I out in a better, more diffusive environment? They do thermotax. They have a way of sensing the difference in gradient, and then also they phototax as a group. So they're using basically everything but gravity to get out. But they're not even trying to get out, so I'm disappointed in them. Like, they just gave up before they even got there.
Okay, yeah. So the lab strains fail in a 3D environment, do perfectly well in 2D. The wild strains do well in 3D. Do they in any way perform less well in 2D?
They actually perform even better in 2D, so it's a great question. They perform, well, I don't know what metrics better would be, but better in that they do this faster, and they make larger fruiting bodies and larger domains. So it's a hint to us that maybe there's a sort of group size that tries to, thinks that can escape certain environments is a hint to us. But yeah, the wild strains appear to be more optimal in basically every way, except they have an inability to eat liquid media. And we've also sort of checked the precursor to this. We've done a few preliminary experiments. So don't quote me if you're a reviewer on a paper. And several years ago, I read X, or I said X. We think the parent wild strain of the current lab strain also has smaller group sizes. We're not sure if that's been selected for over years in the lab of feeding them bacteria on 2D or not. So that's a sort of also a hint to us. There's something about the group size and the group counting that might be at work.
Could you talk a little bit about what we think cyclic AMP is doing? Because in the context of neurons, I have some idea of what its role could be, but I have no idea in terms of the slime mold.
Yeah, so cyclic AMP has a few different roles here. Something that's nice about slime mold as a eukaryotic system is it's very much like us in terms of signaling pathways and everything else. So it's coordinating gene expression through protein kinase A, and so the different pulses of cyclic AMP are helping to coordinate gene expression. That's sort of one thing. It's a really derived behavior for them to be using it outside the cells. It's thought that what happened is that all the slime molds potentially secrete some cyclic AMP late in development. And what happened is that there's an accident that allowed them to end up between them later. But the primary role I think of cyclic AMP is to help them find each other. There's a cool study from a few years ago that actually if you break, this is an argument people go back and forth on, if you break cyclic AMP signaling and then leave cyclic AMP or PKA on so the gene expression is sort of imperfect, that they really were only using it to find each other. And that if they're in contact, they will still be able to put up a structure. It's not perfect. It probably does affect how in 2D. So it probably does affect things like group size. The quorum sensing system is thought to feed back into group size. But I would say its main role here is to coordinate gene expression and then the domain over which you're able to coordinate a group. And so you can imagine if you are able to propagate a signal quite far, that's a thing that cyclic AMP is allowing you to do. Those are sort of the two knobs it has.
So I'd just like a follow-up on cyclic AMP. So in nervous systems, it's G-coupled proteins that trigger the synthesis of cyclic AMP, but what you're saying is the cyclic AMP is released extracellularly. So how does the other cell, how's it work? What's downstream from cyclic AMP?
Yeah, so it's actually a sort of similar setup in that there's a G protein coupled receptor that will sense it in the environment, and it will trigger this whole cascade that leads both to the cell movement, so it will chemotax in the direction it senses that from. It also drives it to generate more cyclic AMP that's then released through a transporter. It will also drive, gene expression and.
So there's a receptor for
there's also
cyclic
a receptor.
AMP.
Right. They don't let it in. They will send it out, and then instead of letting it in, they degrade it, and then in the environment, and then they degrade the degrader. So they actively they pump it out there, and then they get rid of it, but they never let it back into themselves.